In this video, we will discuss about some of the mechanobiological pathways in the case of tumors. So, since this some of this since some of these mechanobiological pathways are related to the glycans, such as this integrase that is decorated by the sugars, and also this cross-linked glycan structures, which occurred because of the overcrowding of the glycans on the cell membrane. So let us first understand how this glycan exists in the extracellular matrix or on the cell surface membrane, and what are the different types of glycans that are usually exist in the mammalian cells. The first type is this glycos uh, glycos glycosphingolipids. So in this case, the glycans are attached to this lipid, as we can see here, and are been further modified by the addition of this salicylic acid. And some of the glycans that exist in this free form are known as hyaluronan, that can be seen here. And thereafter there are glycans which are called GPI anchored glycoprotein. So this type of glycans are attached to this type of glycans are attached to this lipid, and on the other hand the other hand of this GPI anchored glycoprotein is attached to this, and aspargine amino acid of this uh, amino acid of this protein, and this is known as GPI anchored glycoprotein. And there are on the other type of glycans are these proteoglycans. In this case, the glycans which are attached to this transmembrane protein, and has this sulfate and are sulfated uh, in structure nature, such as this chondroitin and heparin sulfates are found on this transmembrane uh, protein, and these are known as proteoglycans. And other part, and the next major one of the major post translation modification, one of the major glycosylation that occurs in the mammalian cells is the N glycans, and there are different kind of N glycans. Out of this first pentasaccharides are common in N glycans, that we can see here. This 2 glycanic monosaccharide and 3 mannose monosaccharide that are common in the N glycan structures. And the next kind of glycans is, and these N glycan structures are found on this aspargine side of the aspargine side of this chain of amino acids. And other part of last part of the glycans is the O glycans, which are found on the serine and threonine site residue of these proteins. And they begins with this galnac and followed by galactose, that can be seen here. So let us start how this mechanicobiological pathways begins now. So the first part is this proteoglycans. Proteoglycans act as a mechanotransmitter, where they transfer where they this proteoglycans helps in interaction with this extracellular matrix, and thereby transfer the force within the cells, and that helps in triggering of the signals such as this RHO, YAP, TAZ, and that will leads to the epigenetic changes. And so these proteoglycans are act as a mechanotransmitter, where they will transport the mechanical forces from the extracellular matrix within the cells, that triggers some of the signaling pathways that I mentioned here, such as RHO, YAP, TAZ. And other part of this mechanobiological pathways is this integrins. Integrins are also act as a mechanotransmitter, and they helps in the transmission. Integrins either on its own act as a mechanotransmitter, or in coordination with the glycans. That is shown here. The mucin is decorated by carbohydrate structures, and also this glycoprotein is shown here. So in all of them in coordination will act as a mechanotransmitter that help in this signaling, that is integrin FAK mechanosignaling. In next part is this growth factor receptor. So how it will work is this silation of this growth factor receptor that we can see here, and thereupon by this activation of this growth factor receptor through this binding of this antigen into this receptor can activate this RAS ERK pathway. So in this case also this silation helps in the activation of this kind of growth factor that triggers this mechanosignaling such as RAS ERK, and this is the combined effect of the glycans and also this antigen that triggers this RAS and ERK through this growth factor receptors. The last one is this is because of the glycans, is because of the overexpression of the glycans around that sometimes might lead to change in the morphology of the cell membrane, and in some cases what it will does is it cross-links because of the overcrowded glycan structure. It cross-links with the extracellular matrix, and especially this sometimes it cross-links with this hyaluronan glycans, which is the free glycans, and that results in the increase in the stiffness of the extracellular matrix around the tumors, and sometimes creates a additional stress, interstitial stress across the tumor, and act as a mechanical barrier, and that helps in evading the tumors from the immune surveillance. And this is shown here, this hyaluronan with proteoglycans and hyaluronans also. Hyaluronan matrix stiffening is just what I have said. The hyaluronan is cross-linked with the overcrowded glycans that are present on the cell membrane of the tumor, and that helps in the stiffness of this cell membrane. That helps in the stiffness of this ECM across the tumor cells, and thereby also act as a mechanical barrier to evade the immune surveillance. So I hope you have understood some basic idea about this mechanobiological pathways and the types of glycans that are present within the mammalian cells. If you need to understand more about this topic, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.